Next, we continue in English with Dr. Not sure if I'm pronouncing it correctly, Dr. Stankic, and she's going to talk about zinc-modified nanocubes as potentially efficient antibacterial agents.
Yes, hello, do you hear me?
Yes.
Okay, perfect. Hello to everybody. So in this talk, I'm going to show you a joint work, a study between our laboratory, our institute, Paris Institute of Nanoscience, and the biology group at the National Institute of Agriculture in France. So at our institute, actually, we are working on fundamental phenomena of different nanostructured systems. More specifically, our group, that is a group called Oxides in Low Dimensions. We are working on metal oxide nanoparticles, in which we are basically looking for model systems which could explain kind of surface reaction of on kind of simple model systems, such as here I'm showing you. I'm not seeing that my cursor. Okay, here. So we are using simple systems, metal oxides such as magnesium oxide, as a model systems to perform different types of surface reactions. Because, for example, magnesium oxide is, with its very simple crystal structure, it is a rock salt, it's a purely ionic compound and insulating material. It's a very simple system on its structure and properties, while when we look at this schematic representation of the surface, we see that it's a quite complicated and complex, so with the different low coordinated surface sites on each different in terms of coordination number, and those kind of sites at the surface, they play a major role in different physicochemical properties of oxides. So we are looking for chemical reactivity, interaction with light. We are also specifically substitute, as I will show in this talk, how we substitute magnesium 2+ by zinc 2+ on this surface, so surface segregation and so on. For this purpose, we are in our lab, we are using two synthesis methods for metal oxide nanoparticles that are both based on the evaporation of metal in an oxidizing agent, so the chemical reaction of the oxidation takes place in the gas phase. And you can see here just a selection of 10 images of different metal oxide nanoparticles, which are produced by these two different synthesis methods. We have, as I have just mentioned, we succeeded in producing a ternary metal oxide nanoparticles, which turned out to, between zinc oxide and magnesium oxide, which turned out to be perfectly one-phase rock salt structure nanoparticles, and with different characterization methods, we have seen that these nanoparticles actually are magnesium oxide nanocubes with the edges only and the lowest coordinated surface sites exchanged by zinc 2+. We have then characterized those nanoparticles and look at the optical properties and interaction with hydrogen at the surface, followed by infrared spectroscopy. And we have observed that those nanoparticles are promising in terms of both optical properties and surface reaction. In any case, that the surface of these nanoparticles is quite different in terms of reactivity, either with light or other molecules, when it is substituted by zinc 2+ in magnesium oxide. So in this respect, we have been thinking what's happening in terms of antibacterial activity, especially having in mind that both parent oxides in this ternary oxides, such as zinc oxide and magnesium oxides, have been already shown to show quite efficient antibacterial activity. But to use nanomaterial for different type of biological or medical purposes, there are key requirements that have to be fulfilled, and those particles have to be well characterized in composition, size, shape. They have to be manipulated without agglomeration, and of course they have to be biocompatible. Having looking at the TEM images and particle size distribution, we see that those kind of cubes are promising candidates, since they are perfectly cubic. It's uniform in terms of size and the shape as well, because they are perfect cubes between 5 and 7 nanometers. So we have then performed different bacteriological tests using two typical model bacteria, Escherichia coli and Bacillus subtilis, and we have followed the growth of the curves by measuring optical density at the 600 nanometers. And we have incubated those bacteria with 1 milligram per 1 milliliter of 3 different oxides, so 2 parent oxides, magnesium and zinc oxide, as well as the zinc magnesium oxide. And we have been following the growth curves of these 2 bacteria. So what we could see is that actually zinc nanoparticles needs less than 1 hour to completely kill bacteria in both type of bacteria, while magnesium oxide and the zinc modified magnesium oxide nanoparticles needs a little bit more time to start the antibacterial efficiency. In any case, quantifying after 24 hours of incubation, quantifying the bacterial viability, we could see that actually zinc magnesium oxide specifically is very reactive against Bacillus subtilis in the same way as a pure actually zinc oxide, being therefore a highly selective also. So the next question we have raised was what about the cytotoxic effects towards human cells? And we did sorry, I was just to mention that we also have performed optical transmission electron microscopy on those bacteria incubated with all of those powders with the concentration I have just mentioned, and we could see the morphological changes of the compared to the pure bacteria that are mostly, that zinc oxide actually mostly affected those bacteria. To do the cytotoxic analysis, we have used HeLa cells, and we did basically just treated those cells with 3 different oxides. Here are the optical images, and you can see basically that only zinc oxide at the concentration of 1 milligram per 1 milliliter is highly toxic because the density and also the morphology of the cells has been quite different compared to the untreated cells. While in the case of magnesium oxide and zinc magnesium oxide, almost no changes in morphology and density. This has been also confirmed with quantification of cell death by staining those bacteria with acridine orange. And we came to the same results that zinc oxide is quite, in a nanoparticle form, it's quite toxic at this concentration, while zinc magnesium oxide and magnesium oxide seems to be quite safe to mammalian cells. In another work, we have then looked at the changes in the morphology and the optical properties of those nanoparticles when they enter into biological media, and we have compared those to water, because all the reactivity and the surface phenomena that we are looking at these materials typically for the surface science, we are working in ultra high vacuum or at least high vacuum conditions. While when bacteria enters, when the nanoparticles supposed to be antibacterial agent and enters then into a biological medium, of course, the interaction with the molecules from the medium should have some impact on its morphology and other physical-chemical properties. We did, again, dissolution of those nanoparticles in, as I said, water, then a typical 2 medium that you use for bacterial growth or mammalian cell growth. I think I said it correctly. And we look at the morphological changes. We already knew what happens from the fundamental point of view. We already did the studies on interaction of water on MgO, for example, and we knew that there is if you imagine those cubes, which are typically magnesium oxide, the interaction with water leads to actually erosion of these corners, and then this increases the concentration of the lowest coordinated the corner ions on MgO, and this is then reflected in absorption in UV spectra, which is typical of these 3 coordinated ions. And this happens here the same, while in a biological media, we could see that there is a decrease in the intensity, which in our opinion was due to the interaction with the molecules in the media. So what was mostly interesting is that when we look at more precisely to the TEM images of those nanoparticles that were dissolved in this RPMI medium, we could see that there is something around the nanoparticles, and we suppose if this is a kind of corona forming, the proteins from the media are adsorbing on the surface and forming the corona. And we did the SDS page analysis, which has showed that from the obtained protein profile, that the most proteins do adsorb on the surface of those nanoparticles, and especially the BSA, which was actually added additionally to this media. We have confirmed this with UV absorption and hydrodynamic radius zeta potential. And then we have checked, because when protein is interacting with another molecule, there is a change in its secondary conformation. We have proved actually by fluorescence spectroscopy, where the typical emission from the BSA was decreasing when we have been adding more and more zinc magnesium oxide. Very similar we got from the CD spectroscopy as well, and we have concluded that there is a weak there is but the weak interaction between nanoparticles and BSC. Then we did also cytotoxicity tests, and we with the crystal violet staining, and we could see that complete cell death was only in the absence of BSA. Again, confirming the interaction with the so I will switch the last paper we have just we did on this type of materials, in which we have studied how the enhanced now zinc concentration in ZnMgO is it improving the antibacterial efficiency, and we have studied a different type of phenomena with B subtilis. I have no time, I guess, to so I will just finish fastly that basically we could see the synergetic effects actually in zinc magnesium nanoparticles, which is highly antibacterial, such as zinc oxide is, and while it is not toxic towards human cells, such as magnesium oxide is. and it's highly selective. We could see that, for instance, nanoparticles cytotoxicity can be governed by the presence of proteins in the biological, such as they are present in the biological media. And the last I will not mention, since I was not speaking about. So I just want to acknowledge the group, biology group I was working with, and all the people at our institute that were involved in this work, especially PhD students and master students. And I thank you for your attention.